more about yourself. Where were you born, and how did you come to pick mycology as a discipline?
Okay, yes. So I won't say when I was born. I was born in Worcester. That's not Worcester, Massachusetts. Worcester in the UK, a small city, a working class family, and we lived on the edge of the city. And so I could just get on my bike, and in 5 minutes I'd cycled out into the countryside and could wander around woods and streams and all this sort of things. And my grandparents were really interested in gardening, so I think I picked up lots about nature from them. None of them had much of an educational background. They all left school at about the age of 14, although I think my dad probably went to night school, something like that. So no great things were expected, but it was just after the war, and my mum and my grandmother, I think they were probably quite bright, and they sort of wanted me to have opportunities that they'd never had. And so they encouraged me all the time. When I was at school, at junior school, I suppose about the age of 8 or 9 or 10, I really got interested in science. I had 2 books for Christmas, and one was on the solar system. Of course, it was the space race and all that, so you couldn't fail to be interested in science. And the other one was on the natural world, and that sort of hooked me. And people asked, what did I want to do when I grew up? And I always said, oh, I wanted to be a scientist or a teacher. So I count myself as really, really lucky that I've managed to do both. And then when I went to senior school, I knew I wanted to be a scientist, but I didn't know much about science. I knew I wanted to work in a lab, and I wanted to work in a path lab. And one of the teachers said, well, why don't you go to university? She said, because you could be the boss of the path lab then. And having sort of rather bossy inclinations, I thought, oh, that sounds quite good. So I started looking at going to university, and I saw that in Exeter they did a degree in biology and mathematical statistics. So my main love was biology. That was despite having some very scary biology teachers, I have to say, and one really nice one. And I was really quite keen on maths, so the two went together really quite well. So I went to Exeter, and probably people will be thinking, oh, okay, that's where she got into mycology, because the head of the School of Biology was Professor John Webster, and hopefully most people know his book, The Introduction to Fungi, and probably also his fungal ecology book. But actually, I didn't get to do much mycology with him, because doing combined honours biology and math, we didn't quite do as much biology, and I wasn't allowed to do the mycology course at all. So I didn't learn mycology from there, although I did do one of his soil ecology courses. But my first real introduction to fungi was when I was living in a flat with lots of other students. It was the basement of a Georgian house, and it was a wonderful Georgian terrace, wonderful houses, except for the student accommodation, where all sorts of horrible things happened, like the sewers overflowed and just things like that. Anyway, one day I tried to open a drawer, and it wouldn't open, and we all tugged on this drawer, and there was what I now know as mycelium. And we tried to move this unit away from the wall, and it wouldn't come. More mycelium. Well, this is interesting, I thought. We looked under the cupboard under the stairs, masses of mycelium. And then these fruit bodies with brown spores. Of course, we now know that it was the dry rot fungus, Serpula lacrymans. And you could imagine that that has now crept along this wonderful Georgian terrace. So that was my first. So I thought, there's something interesting going on here. Anyway, not much more on fungi until I graduated, and I thought, well, what shall I do now? And a job came up, which was a research assistantship on the ecology of wood decomposition. And I thought, that sounds interesting, so I applied for it and got the job, and the rest, as they say, is history.
Wow. So, but that's very surprising answer. But what about then your first experiments in fungal ecology? That must be very, very different than now, isn't it?
Yeah, so this job that I had on the ecology of wood decomposition was really looking at processes. So we were looking at wood decay rate. And this is one of the main things that people did in those days, was to look at, for example, decomposition rate of wood or leaf litter or whatever it was, without very much regard for what was in the wood. And so obviously very quickly I thought, well, there are fungi in here. Why aren't we looking at the fungi? So I said, can we start to look at the fungi, please? And he said, no, we've got the money to work on processes, not the fungi. I was working on wood on the forest floor. Could we please look at wood in the canopy? Because I thought, this is where decay starts. It's those fungi in the canopy. No, we haven't got money to work on that. We're working on decay on the forest floor. And so it went on. Could we look at interactions of fungi with invertebrates? No, because that's not what the project's about. I did need to be reined in, I have to say. But all of these things set me up for the future, and these were things that I looked at. So I ended up looking at processes, rates of decay of wood decomposition, and that is one of the main approaches that I suppose ecologists had, fungal ecologists, decomposition ecologists had in those days. I think a second thing that people did was what I suppose is the start of physiological ecology. People were growing fungi on petri dishes. How quickly did they grow under certain conditions? Did they fruit? Well, not if they were most of the basidiomycetes, but all these sorts of things. And then the third area was perhaps the area which we've come to know much more detail, and that I suppose was the start of community ecology. So people were I suppose 10 or perhaps 20 years before me, started looking at what fungi grow on what. They were listing what was there. And we shouldn't be judgmental looking back and thinking, well, this is rather trivial. You have to remember, this was the infancy of this subject. It was really rather like what the 19th and 18th century botanists and zoologists were doing. So it was really rather looking. Of course, we were seeing fruit bodies, and it started with lists of fruit bodies, and then ideas of succession based on fruit bodies. And I'm sure that people knew without a doubt that mycelium was key to this, and the fruit bodies were not telling the whole story. So people started isolating into culture to see what was there. And of course, one of the main difficulties with basidiomyces, which most of them don't fruit, so the argument was, well, we can't put a name to this. We can list all these other things. We can't put names to this. You actually can get a lot of information from mycelium. Nobles, 10 or 20 years before, was looking at mycelium and tests to try and identify things. Stalpers in the Netherlands did too. I did all my early work on communities in the canopy by isolating from fruit bodies, looking at the mycelium, and then when you isolated mycelium, you could recognize. So you really could, if you were cunning, you could attach names to things. But perhaps most people didn't to start with. I think one of the pieces of work which really stands out in my mind on communities and on succession. We've started to look at succession, following on from what the plant ecologists had done. And there was a really rather good piece of work by Juliet Frankland, which I thought was quite inspirational. She looked at bracken, Pteridium aquilinum, the petioles, and the succession fungi on those, and she didn't just stick to fruit bodies. She made isolations, and more than that, she looked at the change in the chemistry of the bracken petioles over about 7 years. So that was a superb piece of work. And it's only much more recently that we've started to unravel the mechanisms of succession. People weren't really looking at that then, but that's been one of my favourite things with wood, is to, how do fungi change? What are the drivers of change? Is it their environment, or is it interactions? And that's been one of my favourite things.
Yeah.
But you're going to say something now, but I'm going to just carry on, because if you don't mind, there's something I would like to say, that in the mid-1980s, in 1984, Rod Cook and Alan Rayner wrote a book, The Ecology of Saprotrophic Fungi, and I think they really summed up the situation to that point very, very well. And they had the analogy that fungal ecology was rather like chemistry, inorganic chemistry before the periodic table. There was lots of bits and bobs of information, but no structure to it. And they tried to do that in their book. The structure they brought in was from plant ecology, but nonetheless they put a structure there, and they started to formulate ideas around that. And I think that was a brilliant thing. The other thing that they pointed out was that our real problem in fungal ecology was that people were not focusing on the mycelium. The mycelium is the key to the fungal lifestyle, or for most fungi at any rate. And we were not going to get anywhere until we started focusing on mycelium. And Alan Rayner organised a symposium at the University of Bath where those of us who were working on mycelium got together, and I think that was a turning point. You know, we know that mycelium is important, and again, this is something that I've spent a very long time looking at, how wood decay mycelia search through soil for new resources, what they do when they find them, how they send messages back to the rest of the mycelium, how the mycelium then responds, the fractal nature of mycelium, and more recently, really understanding that mycelia are networks, and that is one of the keys to their success.
How do you see the interaction with other microorganisms, bacteria or insects and so on? How do you think that the mycelium is responding to the competition or maybe symbiosis to this?
That's a really good question, and that is one of the keys to ecology. Ecology is, to me, is all about interactions, and one of the things that I'm really fascinated by is how fungi interact with each other. To start with, I've always thought of that as one of the key drivers. So they interact with each other, and they who beats whom is one of the things which drives, very often, not always, very often drives succession. But of course you're right that they don't just interact with fungi, even though many of us have focused on that for a long time. They interact with invertebrates. Invertebrates graze them, and fungi are a major source of nutrition to very, very many invertebrates, either inadvertently when they eat other things like leaf litter, which a fungus is growing in, or in mutualistic symbiosis like some of the termites and some of the ants. But they do eat fungi, and eating mycelium really makes the mycelium behave differently. One of the reasons why mycelia do so well is that they do form networks, and the thing about networks is that there are lots of different interconnections, lots of different ways of getting round a mycelium. So if an invertebrate comes along and chomps through lots of pathways, there are still other pathways there, so the mycelium remains intact. But invertebrates also change the outcomes of interactions. Fungus A might beat fungus B usually, but then one of these grazers comes along who really rather likes eating fungus A, and the whole balance can shift. So it's really rather complicated. Now, if 30 years ago you'd asked me if bacteria were important in wood decay, I would almost certainly have said, no, you don't really find them in wood. But of course, how naive was that? We now know that there are masses of of bacteria in wood interacting with them, sometimes positively, sometimes negatively, probably gaining some of the nutrients or stealing the nutrients that the fungi have got by doing decomposition. And fungi can govern which bacteria are actually present in wood, or indeed, no doubt, elsewhere. So a rather fun experiment that we did not so very long ago was to colonize discs of wood with certain fungi, put them out into the field, and we looked firstly at what other fungi came in afterwards, and so we established that there were priority effects. But it turns out also that the fungi that are present govern which bacteria develop in there. So, there are all sorts of exciting things going on. Another 40 years would be great to work on some of these things, I think, with all the new techniques that are now available.
My next question is, how do you think that the molecular revolution is really influencing or making the new trends in fungal ecology, particularly if I speak about your disciplines?
Well, I think it's obviously a blessing, but I think it's also a bit of a curse. I think it's obviously a blessing in many ways, because we can now put names to fungi which were just perhaps mycelia sometimes. Well, if we use the right database, a curated database, we can add a name. Hopefully, sometimes it's the right name. Obviously, high throughput sequencing is wonderful because it can tell us about lots of fungi that are there, not just the odd few which we can isolate into culture. It can tell us about things which are unculturable, or at least difficult to culture. And so we've learned in lots of ecosystems, for example, the bottoms, the depths of oceans, oceanic crusts and things, that there are lots of fungi there. Obviously, the omics technology is fantastic with transcriptomics, proteomics, metabolomics, and the amazing enzyme tests that there are now, we can find out about the activity, not just what's there, what are they doing? Although, of course, we're still a bit stuck if it's mixed communities. We can't ascribe the roles.
If you have a mixed community and you do with metagenomics, you find quite a lot of bacteria, for example, which you cannot culture.
Mm-hmm.
How do you make the interpretation of any competition or symbiosis and so on? Is that must be very difficult?
I think it is, and I'm not sure that we're really there yet, able to do that, because we can't tell who is doing what in mixed cultures. But, you know, it's a step. You can only do what you can do at the time. And we found this all along. We started off with very simple steps. And I think that people have often thought that fungal ecology is really not very scientific, not very clever. But you can only do what you can do at the time. You know, we've had to get this background information. We've had to get these techniques to come along to find, so we can find what is there, what they're doing in an opaque medium. You know, microscopic organisms in an opaque medium. And these new technologies have also been quite wonderful in that they've brought lots of people into the discipline, I think, that wouldn't have been working on fungi otherwise. You know, you don't now necessarily have to be able to identify fungi to work on fungal ecology. So those are blessings, but on the curse side of things, I think one of the curses is that some molecular biologists, and I'm not just speaking about fungi, know very little about the organisms at all. Or the environments that they're working in. And I think that is very dangerous, because it leads you to pose questions which may be trivial or totally irrelevant. So I think that's a danger. There's also the danger that we've gone back to this listing phase, I think. You know, you can churn out these next generation sequencing things, you get these lists and lists. We've gone back almost 40 years to listing things. We need to take steps forward and to try to answer sensible questions, big questions. Step back and let's have a look. Are there things we don't know which we really need to know, even perhaps simpler things? And I think, yes, there are. We still don't know whether lots of fungal biology that we think we know from stuff we've done in the lab, is this really what goes on in the field? I don't think we necessarily know that yet. Even simple things. You read any textbook, and I'm sure I've been guilty of writing this too. If we're thinking about basidiomycetes, we often imply that in the field they're there as dicaryons or heterokaryons. They've mated. And that happens very quickly. Spore lands, germinates, and they mate. Is that really true? Do we really know that? I don't think we do. One of the most fundamental things. Germination, we know how things germinate on agar, quickly or whatever, what conditions. Do they do this in the field? How long do they survive in the field? Do we really know? I think we don't. We don't even know, and we're ecologists, and we don't know this. If we've got, for example, a branch which is already colonized by a fungus, we know that succession will occur, those communities will change, but how? A spore comes along and lands, a single spore. How does that spore, with no mycelium to start with, then a tiny mycelium, how does that get into wood that is well colonized? You know, there's a fungus in there, done really well, a great fighter. How do those single spores germinate and get in? We don't know. We don't know much about horticology even. That's the ecology of single species. Again, Juliet Franklin did a lot on Mycena galopus, but are there many more fungi that we know a lot of detail about in the real world? I think not. We don't know much about long-term things. Study a system for a long time, you find things out. For example, Tom Bruns's work on some of his systems, he's found out about the spore bank. You could only do that by studying something for quite a long time. We don't do that. And of course, our grant system works against us there. You know, you get 3 years, or if you're lucky, maybe 4-year grants. Ecology goes on for much longer than that. We need to study systems for a lot longer. And we need to collect data over long periods of time too. And I think citizen scientists can come in here, and I'd like to sort of think of back to the future thought really. I started off by saying that people looked at fruit bodies on, well, whatever they were fruiting on, bits of twig, leaves, whatever, and that wasn't particularly helpful. Fruit bodies can be very helpful. We have vast records in Europe and in North America, stretching back for very many years, certainly very good records of fruit bodies for 60 years. Often these records have been obtained by amateur mycologists. And we know that fruit body is only the tip of the iceberg and it isn't telling us everything. But if they hadn't recorded these records, that there are lots of things we wouldn't know. We wouldn't know what climate change has really been doing to fungi if it hadn't been for these records. And perhaps if there's time, I could just tell you a little story about how I got into climate change and fungi.
Well, that's what my next question.
Oh, wow.
Okay, go ahead.
I'm a mind reader too. How about that? So, a few years ago, perhaps 10 or more years ago now, I got a phone call from a professor of entomology at Royal Holloway College, and he said, you know, I don't know much about fungi, but my dad was a keen amateur mycologist, and he said that his dad started recording fungi in 1950, and he and his colleagues from the Salisbury Natural History Society used to go out several times a week, every week of the year since 1950, and they recorded, he identified everything they brought to him, recorded wherever it was from, and he retired and he wondered what he should do, and his son, the professor of entomology, said, well, here you are, Dad, I bought you a computer, digitise every single one of your records, which was on a single card. And then about 6 or 7 months later, he said, oh, I've done that. And so his son then had a sabbatical. He thought there must be something good here, and he started analysing the data. That's when he phoned me up and said, you know, would you look at what I found, this very simple thing, and tell me whether you think it's important or interesting. And the first plot that he showed me simply was the first fruiting date, the average first fruiting date of fungi, and the average last fruiting date. And of course he could get this because his dad and his pals had been out collecting, you know, not just in the autumn, but throughout the year. And it turns out that, okay, until about 1978, yes, there were jiggles. You know that fruit bodies come up different times depending on the weather, but there were jiggles in the line. But on average, nothing much had changed. And in the UK, in the area they lived, there was about 33 days between the first fruiting date and the last fruiting date on average. 1978, things started to change. The first fruiting date started getting earlier. The last fruiting date started getting later. Until now, the average fruiting season is about 77 days, something like that. So it's doubled. And I thought, well, wow, you know, things really are happening here. Okay, this is fruit bodies, but this is telling us something about the ecology of the fungi, and we started doing analyses and showed that this was a climate change phenomenon. And other things that came up were that, well, not surprisingly, that ectomycorrhizal fruit bodies came up at different times than saprotrophic ones. But and also if they were attached to deciduous trees, they fruited differently than coniferous trees, even if it was the same species on these different trees. And so it's quite clear that there are masses and masses of changes going on. Now, when these people started collecting these data, 60 years ago, they didn't know what it was going to be used for. And yet now it's revealed these gems of change. And that's why we need, I think, to do long-term experiments. Who knows in 60 years' time what they might tell us, what they might be used for then.
But if you look to a student who's starting, let's say, in mycology, they rather go to medical mycology or plant pathology. What do you think, how should we tell them that fungal ecology is so important?
Well, to me that's an easy one. I think we as ecologists, and indeed we as mycologists, know that we wouldn't be here if it were not for fungi. You know, I think everybody, any member of the general public, know that plants drive our ecosystems. But perhaps what they don't realise is that every plant is dependent upon fungi, and that we know, don't we, for all sorts of reasons, as mycorrhizal symbioses, feeding the plants. As the decomposers, we break down all the dead stuff. There aren't other things really which can break down lignin to any extent. We'd be up to our armpits in dead stuff if it wasn't for the decomposer fungi. And apart from the inconvenience of having to wade through all this dead stuff, all the nutrients locked up in there
But these are scientific reasons.
Yeah, well,
mostly the student also
this is what. No,
is there a
come
career?
on.
Can I earn money there?
Money? What's money? It's, isn't it the love of the subject? You know, these are scientific reasons. You're right, but this is why it's important.
Of course.
If somebody said to me, I want to be a doctor because I want to save people's lives, I'd say, well, if you just want to save people's lives, you're in the wrong business. You want to be growing plants or looking at plant pathogens, something like that. I don't know how we convince people to be fungal ecologists. Yeah. I think perhaps you have to be born an ecologist. I don't know.
Yeah. Okay. As you know, we organized last year a very interesting symposium for the honor of Johanna Westerdijk, and that was called Leading Women in Fungal Biology, and you were invited, of course, to do this. I think that it's really interesting that there's so many women in mycology, not only in ecology, but in mycology. I hope that will stay, but do you have any advice to a woman who's starting now and say, okay, I want in mycology, or I have to make a choice, maybe I go to another field and so on. Of course, you went into mycology and that was your life, but are there any advices to other female scientists to do that?
I think the only piece of advice I could offer to anybody really is if you get the opportunity to do something that you are really interested in, whatever it is, then go for it. You know, I wake up every morning thinking, oh, wonder what I'll do today. I love working. What will I do today? Who will I meet? What will I find out new?
Okay, if I would give you $1 million.
Oh.
Yes? And you can do only things in fungal ecology. What would you do?
What would I, right, well, after seeing the poster session last night, and I have said that, you know, I think some of the things we're doing are going perhaps the wrong way. There were some great posters, and two sets of them actually I found really inspirational, and I try and see if I could join those groups. One of the sets was that there are these huge experiments in Northern Europe on where they felled trees, and they're looking at the decomposition of these over a very, very long time, and the communities and the processes. I would say, please, can I come and join you and do this? And there was another one by a young postdoc who had made these model systems in perspex, and I was thinking I could make model wood in perspex with lots of little channels in. You see, the thing about perspex is you can see through it. You can't see into wood, can you? And then you could put your fungi at either end, let them go through and let them compete, and you could see what was going on.
And you would need the $1 million.
Oh, I could spend that easily, no problem. We'll move on to doing more climate change things. We've extended our climate change work to Europe. Let's come on, bring in the whole world. Yeah. I'd have no problem spending that money, believe me.
I have still so many questions, but maybe there are questions from the audience. Are there any, let's say, urgent questions you like to ask Lynne Boddy? Yes.
Just quickly about your experience in scientific journals.
Oh, yes. Okay. So I do spend a large part of my life editing. So that's one of the first things. I get up every morning and I go onto the fungal ecology pages to see who submitted what. And I spend a lot of time editing. In fact, every paper that comes into fungal ecology, I do a final edit on. And this is something actually that I learned from David Hawkesworth, how important it is that you put your input in too. And although it's a lot of work, it's a great joy because I have read so many papers on so many different things. Not necessarily always something I necessarily wanted to read, but usually. And I think I've learned so much. I've enjoyed every minute of it. So please, if you are asked to review papers or to join editorial boards, please do it. Science only works by the voluntary efforts of people, well, like all of us. It's really, really important, and it's a great joy. You learn so much. As I say, not always something you wanted to learn, but yes, go for it.
Well, thank you very much.
Thank you. Thank you.